So good morning everyone. How are you? Question papers देख रहे हैं आप लोग? Have you seen all the question papers? देखा है तो how did you find them? समझ आ रहा है कुछ? कंटेंट कम लग रहा अभी. ओके. कोई पेपर जो एक्सेप्शनली यू फाउंड इट टू बी डिफिकल्ट और इजी? इजी का तो मतलब ही नहीं है ना. एनी क्वेश्चंस यू थॉट कि ये मैं कर सकता हूँ? ऐसे क्वेश्चंस थे कुछ कि दिस आई कैन प्रॉब्ली राइट, समथिंग आई कैन राइट, और आई थिंक इट शुड बी अ लॉट ऑफ क्वेश्चंस यू शुड हैव फेल्ट कि समथिंग आई कैन राइट, something I can know. Right? The important thing is कि actually make an effort to see all those questions, because questions से ही indirectly you will be able to understand कि कितना depth में और जाने की ज़रूरत है, ठीक है, and how relevant it is. Like paper 3, if I talk particularly about, so most of it was based on current affairs. There was very few concepts, ठीक है. So in that way for you people now, because you started in February, right? So this is a good time to start linking things with current affairs and try and incorporate that in your assignment answers also from here, right? Because कितने months के current affairs आपने पढ़ लिया है या पढ़ लिया होना चाहिए था? 6 months, ठीक है. So start revising those, ठीक है, which you have already read, those magazines, and now whenever you are writing an answer, try and connect with them with the topic which you have already read, ठीक है. Have you been writing assignment answers regularly? Any particular problem you feel कि हम नहीं लिख पा रहे हैं हर रोज? Okay, so what you can probably do is you can bring that same paper and discuss it with your batch coordinator. If there is a problem you feel कि no insufficient remark है इसमें, तो आप personally उसको discuss कर सकते हो. और what the other thing you can do is, I can suggest is you set up a mentoring session for yourself, ठीक है, and in that you can discuss whatever answers you feel कि you need more input on. Specifically if there is a problem with any one subject you are dealing with कि इसके आंसर में नहीं लिख पा रहा हूँ या इसके जो मार्क्स हैं उसकी कंसिस्टेंसी और वेरिएंस डिफरेंट है, तो आप पर्सनली जाके डिस्कस करो एक बार, ठीक है. यू पीपल हैव द मेंटरिंग ईमेल आईडी? सबके पास है? जस्ट राइट दैट डाउन अगर किसी को कोई दिक्कत है तो. ठीक है. This is the email ID. Whatever issue you are facing in any particular way in this, even if it's with your assignment, even if it's with your evaluation, try and utilize a service like this so that the process goes in streamline. The thing is कि now we are at the end of September. You have 3-4 effective months before you actually go into that prelims mode. ठीक है. उसके बाद ना आंसर लिखा जाएगा, ना मन होगा, ना वो डेडिकेशन से लिख पाओगे, ठीक है. And if your classes are finished, then that is even more demotivating, ठीक है. अभी तो it's like you're coming to the class, you're getting one question every day. You know कि okay, I have to write it, ठीक है, and I can submit it. Once that channel of giving it in hard copy is over, then it becomes even more difficult, ठीक है. Try and finish all pending assignments till the time you have your classes. Okay. Which subjects are left for you? Abhi? Okay. So core main subject जो है exclusive, they are primarily left, right? So once they are over, maybe December, January, जब भी खत्म हो रहा है, उसके बाद it becomes even more difficult. वैसे तो you always have that option of uploading your assignments online, but try and finish whatever is pending at this stage only, because whatever marks you are getting or however you're putting in the content, it is a very good source of revision also. ठीक है, जो आप पढ़ चुके हो, जो आपने February में पढ़ा, उसमें से भी if there is something pending, so now is the time to revise that, put in your assignments, submit it, and focus on the fact कि the first stage जो है assignments की, उसमें you have to focus on content. ठीक है, the difference between writing here and going in at the exam center for a mini test or a whole test is that वहाँ पे time management becomes a more prominent factor. ठीक है, because if you're taking maybe, how much time are you taking to write an assignment roughly? Seriously? I don't buy that, but okay, very good. Good for you if you're doing that. अब थोड़ा सा सच वाला कौन बताएगा? Okay. 20 minutes I buy, 7 minutes I am not going to take. नहीं, गूगल करे बिना भी देखो, इसमें जब आप असाइनमेंट लिख रहे हो एवरी डे, so you have to focus on the fact you're putting in the content and you are structuring it exactly as you feel कि ideally you should be doing. ठीक है, जो टाइम वाला एलिमेंट है ना, उसको अभी यहाँ पे करोगे तो सीख नहीं पाओगे. ठीक है, दिस इज अ लर्निंग एक्सरसाइज. यू हैव टू लर्न कि स्ट्रक्चर क्या है, प्रेजेंटेशन क्या है. When you go for a mini test or when you go for an all India test, that is where the time element comes into play. ठीक है, that will be like 8 questions 1 hour. That is like your standard time for your mini test. उसमें फिर you try and write according to that time. ठीक है, obviously उसमें scope होता है चीटिंग करने का, वो आपको नहीं करनी है. But यहाँ पे inculcate करो कि you have learned everything, you've developed a structure, you have standard ways कि हाँ ऐसा flow chart डाल सकता हूँ मैं, this is the kind of diagram I can draw. Learn this on an everyday basis so that you have so much practice that when you are doing it in 3 hours, that comes automatically. पर अगर यहाँ पे भी time manage करने की कोशिश करोगे, वहाँ पे भी करने की कोशिश करोगे, तो strategy devise हो नहीं पाएगा. So the idea is कि you devise what is the pattern you want to follow, write as many answers as you can, at least in the most ideal way. Till February you should be doing this exercise. By the end of February you can maybe switch over, early March में you can switch over exclusively to that prelims mode. ठीक है, where you are not touching your optional or you are not writing, giving mains tests. But till that time it is very very important that you are writing at least one, if not more than one answer every day. And similar should be your strategy for your optional also. ठीक है, see these questions, give all these questions, take one paper at a time. इस बार का जो पेपर है, paper 1, 2, 3, 4, take 4 days, give yourself 1 hour at least with each paper and just try and internalize what they have asked, what you have covered from that. You can maybe go back to your notes and see what more you can incorporate. कुछ content additional आपको लग रहा है for writing a question is needed, go and add things there. ठीक है, start preparing these papers. ठीक है, then you can go on to prepare other UPSC papers also. But ये जो अभी बिल्कुल आपके सामने आया, you should be very thorough and aware and well versed and linked with it. ठीक है, assignment answers again. Now that you are in the class, make sure you are utilizing all the facilities. ठीक है, जो option मिल रहा है, जहाँ से help मिल रहा है, वो भी अभी बैठ कर नहीं ली, then what is the point of this whole exercise, coming to a coaching institute, paying so much money, right? Discuss it with the batch coordinator if you feel there is some deficiency in your evaluation or in some particular question. Take a mentoring session, bring 10 assignments, discuss that. All right, but do it. Do it till your classes are there. Finish it so that you can actually then move on to the next level. Mini test finish karo, and then when you are doing your probably ideally जो third revision होना चाहिए, that time you should be doing your all India test series. Okay, we'll come back to biology now. Today we are going to take up aspects of genetics हम कर रहे थे in the last class, and biotechnology, and in this class you will also have an assignment question. Okay, because this is the main topic. So what did we do last? किसी को याद है? What was the last thing we did? Okay, we studied blood group inheritance and codominance. Now we are going to talk about a few of our genetic disorders and how they are inherited. The first we are talking about chromosomal abnormalities. So how many chromosomes are there in human beings? Okay. 23 pairs or 46 chromosomes. So chromosomal abnormalities will be associated with the number of chromosomes. So if you remember that there was one cell and in the process of formation of gametes, it divided into two cells with half the number of chromosomes. Right? ऐसा कुछ याद है? We studied this in the last class. Right? Sometimes what happens is that there is one chromosome missing in either case. The homologous pair of chromosomes during cell division, they separate in a way that this can be 22 and this can be 24. One extra pair can go into one gamete and there can be one which will have less, one number of less chromosome. If this fertilizes or if this fertilizes, then the organism formed will have some genetic abnormality, some abnormality as a person because of lesser number of chromosomes. Obviously, if you just think about it, if the chromosomes are less, that means whatever genes that chromosome was supposed to provide, that is not there in this individual. It can still be a functional individual, but its functionality will not be optimum. Similarly, in this case, there is something extra. So that means additionally something will be happening or some function will be restricted. Okay, you understand this? Let's just see some of the examples here. Okay. The first one is Down syndrome. So we have numbered these chromosomes usually for identification. So 1 to 22 plus 1 sex chromosome. You remember this? Autosome, sex chromosomes, right? So in this Down syndrome, the 21 number chromosome comes 3 times. One extra 21 chromosome. 21 number chromosome, we also call it 21 trisomy. An extra 21 number chromosome is present in this disease. It is characterized by small rounded head, partially open mouth, and retarded mental development. Okay, so you have, if you look at the pictures of these children, it's prominently visible from the early ages only. They have a Mongolian kind of a face, a large round, not large, a small face. It is rounded and they usually have their mouth open. Okay. So this disease, Down syndrome, is developed because of an extra chromosome number 21. Okay. The next one is Klinefelter syndrome. This has two X and a Y. So because of presence of Y, this is going to be a male. So this is a male, but because it has two X, it has feminine characteristics. So the main feature of this is that this male is going to be sterile. That means he will not be able to reproduce, and it will have secondary feminine characteristics. Okay, like maybe development of breasts or lesser hair on the body. Such features will be there prominent in this individual. In another case of a female can be the disease Turner's syndrome. अब ये extra वाले we have done, we can have one which has one chromosome less also. So this has X and 0. These are females, but they are sterile females. They cannot reproduce. The reproductive function is not developed. Yes, this is female. जहाँ भी Y chromosome नहीं होगा, it's going to be female only, right? For male, you need a Y chromosome. Only then it becomes a male. Transgender जो हम term use करते हैं usually, they are for people who are mostly have hormonal deficiencies and not chromosomal deficiencies. But जिनमें chromosome जो दूसरा term हम use करते हैं, इनक्स जो होते हैं आपके, यूनिक क्या बोलते हैं? सही बोल रही हूँ मैं? Okay, we can call them transgender also. This condition can be of a transgender. You can have a transgender with something like this. You can have a transgender with something like this. ऐसे ऐसे नहीं, डोमिनेंट रिसेसिव कुछ नहीं है, क्रोमोसोम कम है एक. पेरेंट्स में नहीं, चाइल्ड में. Parent had a normal setup, 46 chromosomes, while formation of gametes, instead of giving 23 equal number, एक जगह 22 आ गया, एक जगह 24 आ गया. Parents are normal only. There is no problem with the parents, but during cell division this has happened, that chromosomes have not equally divided. So it reflects in the child. Parents can be completely normal in this case. Okay. In some of those people, it might not exactly be a chromosomal deficiency, it can be a hormonal defect also. कि आपके क्रोमोसोम्स तो नॉर्मल है, बट हॉर्मोन्स किसी में मेल हॉर्मोन्स ज्यादा प्रोड्यूस हो रहे हैं और फीमेल हॉर्मोन्स ज्यादा प्रोड्यूस हो रहे हैं, उस वजह से उनका कंफ्यूज्ड पैटर्न हो जाता है. नहीं, एक्चुअली होगा वो. इफ यू आर इफ यू हैव फीचर्स ऑफ अ मेल एंड यू आर अ फीमेल, देन ऑब्वियसली यू आर अ ट्रांसजेंडर समवेयर. एक और इसमें दो और कंडीशंस हो सकती हैं, ये देखो. In the feature of this having an chromosomal abnormality is normally that they are sterile. They cannot have reproduction. Okay? That is the main feature. इसमें we can also have super males and super females. Okay? They will also not be normal in their gender types. So one is the XXX syndrome. एक एक्स्ट्रा है, and then we can also have a super male. और इसमें और ज्यादा भी हो सकते हैं कि रेप्लिकेशन हो जाए एंड देयर नंबर कैन इंक्रीज. दैट सेम थिंग कैन रेप्लिकेट मल्टीपल टाइम्स एंड वी कैन हैव समथिंग लाइक दिस ऑल्सो इन अ वेरी abnormal situation. कि एक एक्स्ट्रा तो आया, इट हैज देन फर्दर रेप्लिकेटेड एंड इट इज मोर. दे से मोर द नंबर ऑफ एक्स क्रोमोसोम द मोर tendency a person has towards mental retardation, and the more the number of Y chromosomes, the person has a tendency towards criminal activities. So that is your symbolism associated with X and Y chromosome. Can we go ahead? Okay, now let's talk about gene abnormalities. So genes are part of your chromosomes, ठीक है? And while inheritance, if you remember the last class, we studied कि there's going to be something like a dominant and recessive relationship they have. So we are going to talk about a few diseases which are linked to genes. The first one I talk about is your sickle cell anemia. So we call this disease as autosomal recessive gene defect. So what is happening in this disease is your RBCs are infected, okay? Your red blood cells, they are infected. What happens is that they become sickle shaped. RBCs में क्या होता है main component? आयरन होता है हीमोग्लोबिन में. जो हीमोग्लोबिन है, the capacity of hemoglobin they can hold is reduced because of this kind of a shape they have. And because hemoglobin is reduced, that means its oxygen carrying capacity is also reduced. So, any organism who is suffering from sickle cell anemia, they will have anemia because hemoglobin is reduced and thus the oxygen carrying capacity of the blood is also reduced. Okay? This is the main pattern of the disease. Now how is it is happening? This is a normal pattern of the of an individual, normal hemoglobin you can say, and a sickled organism will have this. Okay? Normal individual, sickle cell anemia. You remember ऐसा कुछ दो factors करके we studied inheritance in the last class? This is your normal individual, this is an individual having sickle cell anemia. Because it is a recessive defect, that means it's only when both of these have, both of these factors are of for sickle cell anemia, it's only then that it will express. Okay? Not otherwise. So if we have a cross between them, then what happens? कुछ याद आ रहा है ऐसा? So if you see, this is the composition of all the children possible. The next generation will be this. This, what do you think? Does this person have sickle cell anemia? Why? Because this is recessive. It is recessive, that means this will be more prominent, this will be expressed, and this is going to be a normal individual, but it has the gene. So we call this individual as carrier. This is the carrier of the disease. This is going to take the disease to the next generation. Okay. So now again, if I have a normal individual and I have a carrier. Okay. So this carrier now goes and together with a normal individual, then what happens? Okay. Again, in this generation also, 50% of the population is going to be carriers. Okay. But if these two carriers come together, then what happens? एक minute. There is going to be one individual which is going to have the disease now, sickle cell anemia. So this is an autosomal recessive gene defect. One, सेम होगा मतलब? हाँ, सेम ही है सारा. हाँ, चारों सेम है क्योंकि दोनों सेम ही चीज दे रहे हैं. जो ये the gamete it is going to give is going to be HBA, same. This is going to be HBS. ठीक है. So there is only one possibility of having only one kind of organism. So all the children are going to be this only 100%. Okay. The next disease we are going to talk about is going to be a sex-linked chromosome disease. Similar inheritance, but with the factor of X and Y playing. So you will be able to understand that if it is prominent in one particular gender. The disease we are going to talk about is hemophilia. sex-linked genetic recessive gene defect. So what is the problem in this disease? The problem is that clotting of blood is not taking place normally in this patient. In hemophilia, clotting of blood is affected, and even the slightest injury to a blood vessel leads to huge amount of blood loss. So the interesting thing is that Queen Victoria had this disease, and this gene is now being transmitted in her family. So it is also called royal hemophilia, or the royal disease. So because it is sex linked, its inheritance we are going to study on the X and Y chromosome. What you need to know is that most of the genes for any disease or anything are located on your X chromosome. Y chromosome has very few genes. Okay. So whenever a gene is there on X chromosome, that gene is going to be reflected even if Y chromosome is there. It is recessive between X and X, but not over Y. अगर Y होगा तो ये dominant होगा, क्योंकि Y has very few genes. But if it is X and X, then this X will be dominant, this normal X. So this individual is going to be normal, this is going to have the disease. So that makes men more vulnerable to a disease like this. Okay. All sex-linked inheritance will have the expression of the X chromosome. Okay. Is this clear? Now let's just see. So we have a carrier female. You understand concept of carrier? We just see this does not have the disease, and we have a normal male. Okay. So what will be their next generation? ये ठीक है? So who has the disease here? What is this? This is normal. This is the carrier. ठीक है, इसको हम बोलेंगे carrier female. This is a hemophilic male and normal female, normal male. ठीक है. So usually we say that females end up being carriers of a disease like this, and males suffer from the disease. Okay. No, there is a gene which is deficient in this. एक gene है, that gene is forming a protein, which that protein is needed for clotting of blood. And that gene is, because that gene is deficient, that means clotting of blood is not taking place. There is no name of the gene. We just say ki gene forming the protein of for clotting of blood. The inheritance of color blindness is also similar. So instead of this, if you have color blindness also, the situation is going to be the same. Sex-linked recessive gene disorder. Write down one question. Let's see if we have followed something here. In context of genetic disorders, A woman suffers from color blindness while her husband does not suffer from it. They have a son and a daughter. In this context, which of the following statements is most probably correct? Both the children, first statement है, both the children suffer from color blindness. Second, the daughter suffers from color blindness while the son does not suffer from it. Third, both children do not suffer from color blindness. And the last one is, the son suffers from color blindness while the daughter does not suffer from it. So what is the cross you've made? Everybody is able to understand, everybody is able to do. Anybody who doesn't understand what is happening in the question? Okay, so what does it say? What is the mother? Mother has a disease, or mother is normal, or mother is carrier? So what will be mother? Recessive disorder, okay. So if a woman has to have a disease, it has to have both those defective genes. What about the father? Okay. So what will be their children? Okay, is this right? Or no? Yes? So how many, what is this? You have to tell me if this is a diseased person or a carrier. What about this? Yes, this is a diseased male. What about this? Yes. And this one? So, all the sons will carry the disease, will have the disease, and all the daughters will be carriers. Okay? So, if they have one son, one daughter, even if they have two sons, two daughters, three sons, three daughters, this is the only possibility which is there. Right? So, all the sons will have the disease and all the daughters will be carriers. Alright? The answer is D. Now we are going to study about nucleic acids before we can talk about specific genetic technology. So for since such a long time we've been talking about DNA, what is happening. Now let's understand what its structure is, what its composition is. The interesting thing is this is the composition of all life forms. Okay? This is the structure which is going to have all the different forms of life. Okay? And basically they are forming your genetic material. So we spoke about the genetic material. What is the genetic material doing for us? Yes. Basic concept, hereditary. Hereditary का मतलब transmission of traits from one generation to the other. ठीक है? From your parents to you, whatever characteristics are being transferred, they are transferred through this genetic material, the basic composition of which is nucleic acids. So nucleic acids are made up of pentose sugar. Remember something like a sugar we studied? Carbohydrates ऐसे पढ़ा था हमने, देश आजाद हुआ था उससे पहले. Remember? ठीक है? Pentose means it has 5 carbons. Okay. It is going to have a 5 carbon sugar. Then it is going to have a nitrogenous base. A nitrogenous base is a cyclic organic compound. And the third thing which this has is a phosphate group. In pentose sugar, there can be 2 types, ribose and deoxyribose. If the sugar is ribose, it is RNA, ribose nucleic acid, and if it is deoxyribose sugar, then it is DNA, deoxyribose nucleic acid. Nitrogenous bases are also of 2 types, purines and pyrimidines. Adenine and guanine here, and there are 3 pyrimidines. Cytosine, thymine and uracil. This is found exclusively in RNA and this is found in DNA. So DNA will not have uracil, RNA will not have thymine. So this base plus this sugar. The structure formed by joining a pentose sugar with a nitrogenous base, we call it nucleoside. And if we add the phosphate group into that, it becomes a nucleotide. These nucleotide units join together to form the structure of the DNA and RNA, polynucleotide structure. So what is happening here is, a nitrogenous base is there, it is attaching to your phosphate group. I mean, this is your nitrogenous base, suppose adenine, sugar, phosphate. ठीक है, इस तरह का arrangement होगा. This is a purine. A purine is going to join with a pyrimidine. ठीक है, so adenine will join with thiamine. Similarly, यहाँ से ये अपना pentose sugar और phosphate group लेकर बैठा, and in between them hydrogen bonds will be formed. 2 hydrogen bonds between adenine and thiamine. और अगर guanine है, so 3 double bonds. Guanine and cytosine will have 3 double bonds. This is fixed. Adenine is going to join with thiamine only. Guanine is going to join with cytosine only. What? This is just one segment. इसके आगे पूरी chain होगी. Backbone होगा phosphate और sugar का, और बीच में ये इस तरह के bonds आते रहेंगे. यहाँ पे मतलब एक और ग्रुप्स ऐसे सीक्वेंस में. So in DNA, this is how the structure is there. There is one chain this side, there is another chain this side, and both these chains are joined together by hydrogen bonds. And then it is twisted in the form of a helix. We call it the double helix structure of DNA. Let's see if we can see a picture. ये प्रोजेक्टर चलाएंगे एक बार? चलाते? Okay. So let's just see a picture so that you can probably imagine this a little better than you are able to do now. This is your structure of DNA. Okay. So these blue colored strips you see, this is the backbone of your sugar and phosphate. Okay. Sugar and phosphate is getting linked and forming a structure like this, the main structure. In between them, there are your nitrogenous base pairs. ये bond जो बना रहे हैं, जैसे this is adenine and thymine. Okay, adenine in one strand and in the other strand thymine is coming. The bond I told you here is a double bond. So in between there is a double bond and they are coming and they are linking these two chains. Okay. Similarly, cytosine and guanine. इस chain में cytosine होगा, इसमें एक में guanine है. This is coming, this will join with this with a triple bond. Okay. And this whole structure is twirled on itself. That is why we call it a helix. Okay, is this clear? So what can happen now is, you remember during when we studied your cell division, we talked about the concept of DNA replication, right? So how is replication taking place? What is happening is कि ये folded है, this is unfolding and strands are becoming separate. Okay, suppose this is one strand. Okay, it has this one strand has opened up now. ऐसे नहीं होगा कि एक strand पे it's going to be only pyrimidines, एक strand पे it's going to be only purines. They can occur in random fashion. So this is there. This has opened up. ठीक है, तो दोनों strands, both these strands have now become separate. Now in DNA replication, both the strands will produce the other part. इसके साथ क्या join होना चाहिए to create a complementary strand? You understand this? This will then both these, यहाँ पे already thymine है, ये अपना एक नया strand बना लेगा, ये अपना एक नया strand बना लेगा. So DNA will be replicated in that sense because of complementarity. कि this knows कि इसको किसके साथ join करना है, this knows इसको किसके साथ join करना है, ठीक है? Cytosine will join with guanine, ठीक है, thymine, adenine, ठीक है, this again cytosine will है, ठीक है, तो इस तरह से the arrangement, whatever the sequence can be anything, but they will create complementary strands and DNA will be replicated, and then this DNA, if you remember from cell division, it will go into two different cells which will have the same DNA as the original parent, but now we have two daughter cells. Can you connect? Is this confusing? Are you okay with how this is happening? Another condition is possible कि DNA की जगह अगर इसको RNA बनाना है, इसी strand को there is a small segment of DNA which we want कि इसमें से whatever protein it is coding for, that should be created. But proteins are formed by RNA, ठीक है? Where are proteins formed? Any organelle you remember? Ribosomes, right? Ribosomes. पर वो RNA के through हो रही है. So this can also, a small segment of DNA can also create its own RNA also. अगर इसको RNA create करना होगा तो क्या करेगा यहाँ पे? Yes, ठीक है, uracil, hair. What about this one? ये तो ऐसे ही रहेगा ना, पर ये तो एडेनिन कर सकता है ना, ये तो कॉम्प्लीमेंट्री के साथ करेगा ना ज्वाइन. व्हाट इज कॉम्प्लीमेंट्री टू थायमीन? Adenine is there in RNA also, right? Only this is not there, so this will be uracil. What about this? ठीक है. So इतना इसने RNA strand बना दिया, and then this again will then go for protein synthesis. ओके, सो डीएनए के ऊपर डीएनए भी बना सकते हैं हम. डीएनए के ऊपर आरएनए कैन ऑल्सो बी फॉर्म्ड, एंड दिस आरएनए विल देन गो एंड एक्सप्रेस इटसेल्फ इनटू प्रोटीन्स. वील जस्ट राइट अ फ्यू थिंग्स. Okay, write down. The nucleus. Where is DNA present? The nucleus of a living cell is responsible for transmission of inherent characters also called heredity. Nucleic acids are responsible for transfer of traits from parents to offsprings, thus making them the genetic material of all life forms. ये लिख लिया हुआ है ना आपने, पेंटोज शुगर, नाइट्रोजन बेस, everybody is written that? Okay, write down. The DNA molecule or the DNA is a double helix structure made up of two complementary polynucleotide chains. DNA is capable of self-duplication during cell division and thus identical DNA strands are transferred to the daughter cells. So that concept of DNA to DNA and RNA to protein I talked to you about, that is actually technically called gene expression, right? So we have some genes, but those genes have to actually express themselves to lead to some function. Okay. So two processes are involved here. The first is transcription. And the next one is translation. This is taking place in the nucleus, and this is taking place in the ribosomes. This is DNA forming RNA, and this is finally your RNA translating into proteins. Do you think of any other place, any other organelle? एक तो ribosomes are kept in your cytoplasm. Anywhere else we have ribosomes? No, not in mitochondria. Any other organelle, if you remember, which had presence of ribosomes? ऐसा कुछ याद है? Rough endoplasmic reticulum is associated with protein synthesis. Why? Because it has ribosomes, and in this place, the ribosomes, it will happen that they will, from RNA, they are going to form proteins. So amino acids ke liye, usually triplet code is there of this. Like ACT is going to be one amino acid, CTG is going to be another amino acid, like this they are coded. So on that long change, whatever coding is there, accordingly the protein is formed. So you can also just note down ki amino acids are kept in or identified through triplet code. So if there is a slightest error, that means a different protein will be formed. So you understand this? अगर ACT की जगह ACD हो जाए, so the protein entirely will change. That is why the defect of all these diseases. Okay, this has to be exact for creation of that particular protein or that amino acid. One more concept associated with this, which you all know about, is your DNA fingerprinting. So what do you know about this? सुना है किसी ने? DNA टेस्ट करवा लो, paternity dispute के लिए करवाते हैं usually. What is it? Anything you know about this? सुना है? So anything? Okay, criminal identification के लिए use कर सकते हैं. करते क्या है basically? Okay, so probably we'll identify something on the sequence and see if it is that person or not. So इसमें क्या होता है? Obviously this DNA, where is it coming to us from? From our parents. ठीक है? So that is where we will match. There is something we call as VNTR, variable number tandem repeats. So what is this? This is a repetitive sequence. ठीक है? There is one particular sequence in all which we get from our parents, which we inherit from our parents, which is repeated. मतलब DNA का अगर ये लंबा सा सेगमेंट होगा तो उसमें कुछ पोर्शन ऐसे होगा जो बार बार रिपीट हो रहा है. Suppose this is the area which is repeated. This is your VNTR. This we usually identify in this technique. How this is identified? Like we get a DNA. So where can we get your DNA from if I have to take your DNA sample? Almost from anywhere. Okay, any part of your body we can use for, I mean even the slightest part like your saliva. Okay? So everything you drop anywhere, it's going to be very dangerous. So if you have to go to a crime scene, you have to make sure that there is no hair fall. You cannot leave any hair strands behind. Hair, blood, skin, bone, saliva, everything. छोटी से छोटी चीज में भी बहुत खतरा है. फिंगर नहीं, फिंगर टच से नहीं हो सकता. नहीं, वो दैट विल बी बायोमेट्रिक, दैट विल बी एबल टू, वो फिंगर प्रिंटिंग ही हो गई फिर. एग्जैक्टली ठीक है, दैट इज डिफरेंट. बट नॉट डीएनए, वी कैन नॉट गेट डीएनए फ्रॉम दैट बिकॉज इट्स जस्ट एन इम्प्रेशन. ठीक है, बट अदर देन दैट एवरीथिंग, सो व्हाट दे डू, दे पिक इट आउट. उसमें से डीएनए कैन बी. Toothbrush में actually what happens is some amount of your saliva or some dead cells from your gums can come over that, and then yes, that can be used. So be careful where you are brushing. Yes, sir? So what we are doing is we are taking up that, we are picking out DNA from that, we are isolating, and we are DNA को क्या करेंगे, अब इतना बड़ा strand है, we are going to put it and break it in pieces. ठीक है, in different pieces we are going to separate them. We are going to have some markers and identification किस segment की करनी है, इसकी. Okay, we identify this segment. How we are going to do? Again, इस पे दिमाग लगाओ. If we are finding this, suppose I am finding this segment, that means I am going to prepare, let's make it DNA only. I am going to prepare this. This is, I think that this is what I have to look for. Then I'll make a segment like this and I'll send it in that whole range जहाँ पे DNA segments पड़े हैं, which I have broken down into smaller pieces. If this goes and joins, that means this is the sequence. इसका हमें पता है, अगर इसके साथ join किया तो मतलब ये होगा. That we can calculate, this simple logic we have, right? If this is calculated, then we say this is a match. This is how we identify. तो जो VNTR हम find कर रहे हैं, उसका complementary stand we are going to prepare and we are going to send it with a marker, and we know कि if it is joining, that means that identification has taken place. Okay? Simple. This complementarity जो है, this is the basis of this test also. The process of us joining one segment to the other, that is called hybridization. Just joining together of fragments of DNA. Hybridization from this VNTR. If I have to suppose, for example, if I am looking for a paternity dispute, I have a DNA sample from the father, I have a DNA sample from the child, I will identify the father's VNTR, okay, I'll see what sequence is there, and complementary to this, I am expecting that this segment will be there in the child also. So complementary to this, so suppose this sequence, like I said, it is ACTG, then I will make a probe ठीक है, an investigative DNA I will make which will have TGAC. ये मुझे ढूंढना है, पर ये सीक्वेंस अलग हो सकता है ना हर इंसान में. जरूरी नहीं है ना किसी में ए, ये एक, दिस इज वन ऑर्गेनिज्म, देयर कैन बी अनदर ऑर्गेनिज्म विच कैन हैव TGAC. जितने मर्जी बना लो इसमें, and there are like millions over in one DNA strand. Yes, this sequence is different in every person. Some of that sequence, see there are 4 variables, ठीक है, which we have to use. They can be arranged in any number of fashion you want across the length. वो hundreds और thousands के नंबर में एक DNA strand पे पड़े हैं अगर, तो वो सीक्वेंस कुछ भी हो सकता है. You can say like fingerprints, yes, this sequence is unique for everybody. This sequence of your nitrogenous bases, this sequence of nucleic acids, it's different. And that unique feature we have to identify. ये एक just easier तरीका है, आप इसको भूल भी जाओ चाहे, if you want to keep it more simple, then you can just think about the fact that there is a unique feature which we are looking for, ठीक है, एक unique sequence जो हमने match करना है कहीं से. We have to match it from somewhere. In a paternity dispute, suppose it's going to be matched with one of the parents. So we have that from the parent, we know कि what part of the parent would have gone to the child. We make a complementary stand for that. ठीक है, अगर वो उसका ही बच्चा होगा, ठीक है, if this is the child of that same person, then this will go and match somewhere in that whole strand of DNA which we have broken down into smaller pieces now. If this goes and joins, then we know कि this is present. ये तो सही है. If I have TGAC and this goes and joins somewhere, so I can conclude कि ACTG is there. Are you following this? ठीक है, so if we have identified कि this is there, that means the match is there. हमें पता है हम किस चीज के साथ मैच कर रहे हैं पहले ही, that has already been identified. Okay or not? हर किसी के लिए अलग अलग होगा, ये एक हो ही नहीं सकता. पेरेंट्स से भी कुछ सेगमेंट सिर्फ हमारा सेम होता है क्योंकि हम वी आर गेटिंग इट फ्रॉम बोथ द पेरेंट्स ना, दैट इज वाई. So some of our sequence will be matching with our father, some will be matching with our mother. But overall it is not the same, right? Because we are a mixture of both of them, right? So this sequence is different, this is unique, and that is how we can be, an individual can be identified. Okay. Multiple? हाँ, multiple sequence. हम एक DNA strand को we are going to break up in a lot of pieces, many number of pieces. In different segments. So basically what you need to know is कि how and where we are taking the sample from, ठीक है, your blood, hair, skin, bones, ठीक है. What we are doing then, we are isolating the DNA, we are breaking the DNA, and then we are hybridizing it with known segments to see if the match is there. Then what you have to know is its application. What can be the application? One, we have established paternity disputes, then crime scenes or forensic studies, right? Anything else you can think of? No, not disease. This is usually for matching. Maybe something like we have unclaimed dead bodies. We come across them maybe in an excavation, that kind of a site. Or racial profiling. Maybe or anthropological studies may we can identify ki which communities or which tribes are matching how much with the other tribes. Such kind of anthropological studies also can use something like a DNA fingerprinting analysis. Government is actually focusing, yes you can say, and also they are trying to regularize कि इसका misuse भी ना हो, because this is considered to be an infringement of your privacy also. You giving out your hair and they having a sample of your DNA and keeping a record of that is in a way an invasion of your privacy. So that is the issue. That is why they are trying to regulate it. Okay. Okay, we'll start with biotechnology after 10 minutes. Okay. फिर से ओके, 2 पेरेंट्स हैं A एंड B. अब इनका एक चाइल्ड है C. अच्छा, किसकी करवानी है? ओके, A ने अब मैरिज किया किससे? D से. अब चाइल्ड होगा E. इनका DNA कुछ सेगमेंट में मैच होगा. यस, you will be able to identify that they are siblings because this person is getting some DNA from A, this person is getting some DNA from A, right? So there is going to be some match will be there. Yes. So if we check for their DNA, we will be able to identify that they are siblings or related to a common parent. Yes. Okay. Okay, so we're going to cover some aspects of biotechnology now. What do you understand by biotechnology? Bio plus technology, okay. See, bio, what does bio mean? What is the context of life? Okay, so any living organisms which we are utilizing, ठीक है, technology means कुछ हमारे लिए उसका use होगा, ठीक है, which is, we are changing it in some form to be utilized for human beings, for human life. That aspect is covered in your biotechnology. You can write down in one line also. It deals with the techniques of using live organisms or enzymes to produce products and processes useful to human beings. So we have studied the basis of genetics, basic of genetic material, ठीक है, DNA है हमारे पास, RNA is there. Now here what we are going to do is we are trying to make some changes in these structures so that we can produce some useful products or some useful forms. Why do you think any changes is possible in this or any manipulation is processed? Anything you can think of? See, this is common to all life forms. Because it is common to all life forms, that means it is possible for us to subtract, add, join, create new forms. ठीक है, we can have a bacterial gene inserted in a human gene. Why? Because basic sequence of the DNA is the same, right? DNA जहाँ पे भी होगा, it's going to be that basic nucleotide structure because it is chemically same, so it is possible for us to have a modulation being done in this. Different types of technologies or different usages are there of this exercise of modulating this. We call it genetic engineering. And we are going to study all the different cases, that what we are doing in different cases and how it is helpful. You will keep coming across all of these almost every month in your current affairs portion. So try and understand the basics very carefully so that you are able to understand those kinds of news. The first thing we are going to study is recombinant DNA technology. Before that, let me just talk to you about the types of biotechnology first. वैसे तो बाद में भी कर सकते हैं, पर चलो यहीं पे पहले करते हैं. So different types of biotechnology would means where we are using them or where the application is taking place. The first one is green biotechnology. Where do you think this will be used? Agricultural based or plant based, horticulture based, such things. So you can just say agriculture primarily. Okay. Blue, yes, marine life, ठीक है, all the life forms associated with water, that will have your blue biotechnology. Red is going to be for medical applications. Okay. Medical applications of biotechnology. Then we have white and grey. White biotechnology for your industrial application. And grey biotechnology for environmental use. Grey for environment, environmental application. So whenever you keep coming across any examples, you have to think about categorizing them in this. Suppose in agriculture, we create a crop which is resistant to pests. Okay, we create a crop which is resistant to pests and thus can survive longer in a particular environment. That becomes a part of your green biotechnology. Like your most common example is your BT cotton, BT mustard, all these crops. Okay. Then blue mein, what do you think we can do in marine? Corals are there, but okay, suppose I have a particular fish variety and I engineer its DNA in such a way that it has more nutrients or maybe more of a particular vitamin, right? And those forms we start cultivating. That can be an application of blue biotechnology, right? What about red? What about the medical application of biotechnology? Yes, clones can be one way. Yeah, okay, insulin production. Maybe we can have a bacteria in which we can insert a gene that bacteria starts producing insulin now. We take that insulin, we bottle it and we start selling it in the market. That can be a medical application under your red biotechnology. What about industrial usage? Okay, dyes can be one thing. Food processing may, okay. Use of? Okay, different molecules, or maybe see, suppose I am creating a detergent, ठीक है, and I have an organism which is producing enzymes for digestion of fats, ठीक है. So I take that gene, I insert it in a different organism and I start producing that enzyme. That enzyme I mark it as a part of a washing powder, ठीक है, उसमें वो enzyme incorporate किया. So now when that washing powder is acting on the clothes, then any fat or grease or oil marks on the clothes, that can be removed easily, right? This way we can have a medical an industrial application of biotechnology or products of living forms. Similarly with environment also, आप ये सब पढ़ोगे जब environment भी पढ़ोगे, like there is the most common example is that of an oil spill, ठीक है. So oil spills wherever they are on water bodies and we have to remove that oil. It is a very tedious task to remove that oil from a water. If suppose an oil tanker is going or wo, due to some accident that oil has spilled. Now that oil, what it's going to do is it's going to kill the organisms there. So what we will do is we will develop a strain of bacteria or any organism which can digest oil. Like एक बहुत common example है oil zapper का. So what this, you'll read about this in your in environment also. So what is this? This is like 5 bacterial strains which we have brought together, and what it does is it digests your oil. Okay, that is an application of biotechnology in the environmental aspect. So in the gray part here, right? So after this type, let's come to your recombinant DNA technology. What does this word tell you? Recombinant DNA. What do you understand if you just try and break it out in your basic English? Recombinant का क्या मतलब होगा? Okay, yes, it's a combination of DNA. So that means a DNA we are getting from 2 organisms or even more is possible together, and then we are utilizing it for some purpose. So let me talk about the case of insulin. Okay, I am a pharmaceutical company. I want to sell human insulin. Okay, so that means I need some form to produce insulin. So what I am going to do is I am going to take a healthy organism, a healthy human being. उसके अब कहाँ insulin, where is insulin produced? Yes. So I am going to take a cell from the pancreas, the insulin producing cells of the pancreas. I am going to take that cell, and from that cell I am going to extract the DNA and I am going to extract the gene for insulin production. ठीक है, अब मेरे पास insulin gene आ गया, right? What I do is I take a bacterial cell now. After this, after I have isolated this gene, I am going to take a bacteria which is going to act. So bacteria is eukaryotic or prokaryotic? Prokaryotic. What does that mean? Nucleus is not there, a well-defined nucleus is not there, DNA is present in the cytoplasm. ठीक है, तो along with this DNA, some bacteria also have a circular DNA along with it, which we call plasmid. Plasmid is your single circular DNA which is present in some bacteria. It is independent of the DNA of this nucleus or of the nucleoid region. So now there are going to be 2 kinds of enzymes. One we are going to call endonucleases or nucleases, and the second one is going to be ligase. So if we have to join, cut and join, it is basically cut paste. Okay, we are doing cut paste of DNA. So for cutting, this is the enzyme we need. This, you can say this is the scissors which is going to cut, and for joining, this is the enzyme ligase. So यहाँ से भी when we have to remove, we will need a specific endonuclease which is going to cut this gene off. ठीक है? And what we are doing is, we are taking this plasmid from here, in this particular case, and we are taking that gene for insulin, and we are inserting it here. In the plasmid. Okay. So some part of a human gene is now joined with the plasmid of the bacteria. Okay. Then we make it a point that this replicates. We grow this in number, we insert it back into the bacteria. So what? Now this bacteria will start expressing this gene. What is the role of this gene? Yes, so that means this bacteria now will start producing human insulin. This has become the recombinant DNA, we have inserted it back in the bacteria. So this is now going to start producing insulin. यहाँ से जीन ले लिया हमने, यहाँ पे एक सेल था, इसके डीएनए में से हमने सिर्फ जीन निकाल लिया. Okay, we have gene. Then we have a plasmid here. This is a single circular DNA here. इसको उठाया, इसका कुछ सेगमेंट हमने काटा और उसकी जगह हमने this gene. ठीक है, for cutting we have endonuclease enzyme, for joining we have ligase. So with the help of ligase we have joined it. This plasmid is now in the bacteria. ठीक है, so this gene will start expressing itself, and when it is expressing, what is its role? To produce insulin. That means this bacteria will start producing insulin. Bacterial traits, it is already there because it has that, but additionally because we have inserted this gene, that means it will now start expressing this also. So it will start producing insulin. That insulin I can use, bottle and sell in the market, because that is human insulin. It has come from a human being's pancreas. Okay, simple process. Okay, you pick out a gene, you take another organism. इसको हम बोलते हैं, जो plasmid होता है, we call it vector. Because it carries gene to the host cell. See, now this host cell can be some other cell also, not necessarily that same bacteria. We can incorporate this in some other organism also. Okay, so whichever organism this is sent, this vector goes in, that organism will start expressing that gene. In this case for insulin, otherwise whatever gene we wish to add, that gene we can add, and the function of that gene will start showcasing. The basic idea which I wanted to stress upon is that this is possible because the arrangement is same. DNA structure is same for all organism. If it's a bacteria, if it's a human being, because the structure is same, that is why they are able to chemically join. Okay? If that structure was not same, then they would not have joined chemically. They can join because the same sequence, the same basic molecular basis is same. हाँ, हेलिकल स्ट्रक्चर होगा, जहां पे भी डीएनए होगा वही उसी तरह का होगा. देयर इज गोइंग टू बी अ नाइट्रोजनस बेस, देयर इज गोइंग टू बी अ फॉस्फेट ग्रुप, देयर इज गोइंग टू बी लिंकेजेस between the groups. It is all going to be the same everywhere. Bacteria ho, human being ho, cow ho, pig ho, whatever. Sequence is different from all the things, but at least chemically they are same, so that is why they can join. जो भी sequence होगा, according to that the traits will be expressed. We have introduced a sequence which is there for insulin production, so it will start producing insulin. Sequence will be different for all organisms. For you and him also sequence is different. But chemically it is same. The chemical composition of nucleic acids remains common. That is why they form bonds. They can form bonds whenever we want them to. All right? Okay. So this is the common basis of recombinant DNA technology, which we are going to, which again has applications in all those areas. ठीक है, जो हमने different areas किए पढ़े, उसमें यही concept involved है, that we have a gene, we pick out the desired gene from an organism, we join it with another organism, that DNA becomes recombinant DNA, that starts expressing whatever traits we want it to express, right? And then it can grow and further reproduce. Okay. Okay. Can I come to the next concept after this, after recombinant DNA technology? Okay. Your next concept is going to be animal cloning or cloning in general. The enzymes remain the same in every case whenever we have to cut something or we have to remove something. Nuclease for cutting, ligase for adding. Okay. What do you understand by the word cloning if I use it? Okay. Identical copy. Okay. So remember we studied a form of reproduction also where genetic material remains same. Okay, in mitosis you can say that's a cell division, but in all forms of asexual reproduction also the material remains the same. That is a technology which we are going to use. Again, because it is animal cloning, a clone is formed, there is going to be a single parent here. Okay. There is, so the cell we are going to pick out is an ovum. Okay, ovum or egg is the cell which we pick out usually. So is ovum ko, there is a cell, there is a nucleus, right? What we do is, first step we do is enucleation. Enucleation means we are going to remove the nucleus. So in an organism, you remember something like this, 2n, n, haploid and diploid forms. What is the form of the ovum? Ovum is your female gamete, ovum or egg. Ovum and sperm, remember? It cannot be diploid, it is going to be haploid. जो gametes हैं, वो हमारे n हैं. They have half the number of chromosomes because an ovum is going to come together with a sperm which will also have n, and then they will form a normal organism of 2n, of diploid organization. Can you connect this? Right? Meiosis has taken place. The chromosome number or the genetic material has been reduced to half, and this ovum now has n. Haploid set. Okay. The parent here, this is a sex cell, okay? So this, we are going to take a normal somatic cell here. Any cell of this person, we are going to take a normal cell and which has a nucleus. What will be the composition here? Haploid, diploid of a normal body cell. Normal body cell is diploid. ठीक है, 2n होगा यहाँ पे. इसका हमने न्यूक्लियस ले लिया सिर्फ. So we have an enucleated ovum We have an enucleated cell here and we have a nucleus here. They come together. We have a cell which has the nucleus of this organism in the ovum of another organism. तो ये अभी यहां से 2n ही है. Then what we do is we chemically trigger this to start growing. It will not immediately start growing. Here what happens is कि zygote is formed. So this zygote will start growing into an embryo. ठीक है, further for the formation of the embryo and then eventually the baby. But ये तो क्योंकि ovum ही है तो this will not automatically start growing. We have to induce it to start growing, start dividing. Once this start dividing, what we do is we take this after it has achieved a substantial size and we implant it in the uterus of a female. There it starts growing into a baby and this organism is going to be exactly like the parent. We are taking a somatic cell, we are transferring its nucleus into an enucleated ovum, making it grow in the lab, then again transplanting it into the uterus of a female, where this organism will come out through a normal birth process. Okay. Is this clear? Are you able to understand how this is happening? The technology we call this one is somatic cell nuclear transfer technique. This is a somatic cell. We are transferring its nucleus, making it grow, transplanting it into the uterus. Okay, and then it is growing as a baby. Any confusion? Anything you're not able to get here? Zaruri nahi hai. It can be any other female also, right? So there is this.
There is a mixture of mitosis and meiosis. So this is a somatic cell. This comes from the mitosis process and this one comes from the meiosis process. And how mitosis and meiosis?
Mitosis and meiosis keep taking place at every stage. It's just normal cell division here. यहाँ पे actually you don't have to consider any cell division. I'm just picking out a cell. Okay. So I pick out a cell from your body. Any cell I pick out. ठीक है. एक cell ले लिया. Now I'm going to make a clone. I'm going to make your clone. So what I will do is I will take an egg from a female. ठीक है. From any female. I will take out the nucleus of that egg. From your cell I will take out your nucleus. I will put it in her cell. जो खाली सेल है मेरे पास बिना न्यूक्लियस के उसमें दूंगी. आई एम गोइंग टू ग्रो इट इन द लैब. वंस इट स्टार्ट्स ग्रोइंग आई एम गोइंग टू ट्रांसप्लांट इट इन द यूट्रस एंड आफ्टर दैट नॉर्मल gestation period which for human beings is 9 months a baby will come. That baby is going to be your clone.
How the normal cell can work? We need some little cell like sperm or any sperm.
No, you need a sperm if it is n. ठीक है. जब आधा क्रोमोसोम होगा then we will need another part. This already has 2n. It already has a full set of chromosomes, so we can grow it. We will chemically trigger it to start growing. That is the process. This is happening chemically in the lab. Yes, exactly. See, unless and until whatever environmental variations are there, that will be expressed in that organism, but dimag mein, form mein, everything will be same. Yeah, everything, IQ, everything will be same. So if you take that baby, like his clone, we have that baby, but we actually have exposed him to a lot of sunlight and gradually maybe he ends up getting more tanned. So such environmental factors can have their variation, but otherwise everything is going to be like him, other than the difference of age. Okay. Okay. So the concept of surrogacy or surrogate can also be understood from here. So the female or the mother who actually bears the baby has nothing to do with the genetic composition, right? So the female who is holding the baby and who is actually giving birth to the baby has no input of any of this. She has nothing to do with the DNA. She is just holding the baby and basic nutrition is being provided to that baby from her blood supply, otherwise nothing. In genetic composition in parentage, it is exactly his baby. It is just he is the single parent. This person has nothing to do with it. This is the concept of surrogate. Otherwise also anywhere where you coming across this term as the surrogate mother or surrogacy, that means कि genetically कोई लेना देना नहीं है. They're just taking the womb of that mother for birth. ठीक है, हम ऐसे भी कर सकते हैं. In this case also, we take an egg from a female, we take a sperm from a male, we fertilize them in the lab. Once it starts growing, we transplant it in that surrogate and that baby grows there. That is the concept. You've read about surrogacy, right? It's been in news. Surrogacy bill. ठीक है, वो यही चीज है कि parents are other parents, but maybe this female does not have an ovum strong enough to hold that baby or to hold on to that delivery because of any pathological problems she has. So there can be another person who can actually have that baby, hold that baby. It is transplanted in the uterus and then that baby grows its normal course. ठीक है, पर अब इसका अगर DNA analysis भी करेंगे, even if the baby is born from that mother, but once we start analyzing the DNA or if there is a paternity dispute, then it will be very clear कि उसका कोई लेना देना नहीं है, because she doesn't share the DNA with that baby, right? ठीक है? Can we go on to the next concept? Okay. So, along with this cloning concept, अभी एक new concept आया है. This is a single parent. Now we have 3 parent babies also. Have you read about that? Okay. So let's talk about that now. Okay, so what have you read? What do you understand by this? This has been in news recently. क्या है ये? Anybody who understands this? Yes. Okay. So, yeah, you're absolutely right. Which other organelle in the cell have DNA? And animals की बात हो रही है. Yes, mitochondria and nucleus. ठीक है, basic genetic material in your nucleus. Other than that, your mitochondria also has its own DNA. ठीक है, we call it mitochondrial DNA. Okay. This DNA in the mitochondria, वैसे it has very few genes, right? Somewhere around 37 to 40 genes it has. I think 37 is the number. But sometimes there is a possibility that this DNA can showcase some diseases. ठीक है. In the mother, this mitochondria, first thing you have to know is inherited exclusively from the mother. In a baby, it gets nuclear DNA or the basic genetic material from both parents equally, but this mitochondrial DNA, it gets exclusively from the mother. So that means if this DNA has some disease, it is going to be expressed in the child. Okay. There can be a number of diseases which have been associated with mitochondrial diseases, and like Parkinson's disease is one such example. ठीक है? Yes, diabetes also has some linkage with this mitochondrial DNA. ठीक है? So such diseases, if we have identified them to be a part of our mitochondrial DNA, they are sure to be transmitted to the child. So Now we have understood कि this is happening, so we are going to find a way out. What is the way out? The way out is that this DNA, this mitochondrial DNA can be brought in from a third parent, from a third female. So what we will do, we will find a donor. Okay, this is a female, and again ovum or egg, but we don't need her DNA, we don't want her nucleus, so it is going to be enucleated. Enucleated means the nucleus is not there but the cytoplasm is there, so mitochondrial DNA or mitochondria we will be able to inherit. दूसरी जो आपकी नॉर्मल मदर है उसके एग से क्या लेना है हमें अब? न्यूक्लियस, राइट? Again that similar process, but this is not a somatic cell transfer because this is a gamete. ठीक है, this is not a normal cell, this is your sex cell, egg. इससे न्यूक्लियस लिया, ये खाली सेल है, this is a normal ovum, this becomes a normal ovum. This ovum then again with your sperm it joins, we have a zygote. Again because this process is happening in the lab, this will start growing, then we will again insert it back. We can, in this case there is a possibility of inserting it back in the mother also. If you want to do it in another surrogate, that is also possible. Anyways, ठीक है? This is also again fancy cut paste. इसका माइटोकॉन्ड्रिया सही नहीं है, वो निकाल दिया. इसका सिर्फ माइटोकॉन्ड्रिया चाहिए था, वो ले लिया. स्पर्म तो जो फादर है वो तो है ही है नॉर्मल, and then this baby can grow. What do you think of the genetic composition of this baby? It is from how many parents? 2 or 3? उसमें भी तो genes हैं. See, the genetic composition of this baby is from 3 parents because this DNA is also there. But the point is कि because the number of genes are very few, so it does not make a lot of difference. जो main inheritance, main traits होंगे, that is going to be of those 2 parents only. Okay, so this is your 3-parent baby, or it is also called mitochondrial replacement therapy. You can write that down also, mitochondrial replacement therapy. Side effects. See, as such of all these things, there are no side effects if you feel ki some form of abnormality if you are asking about in the baby. Nothing like that is there. It is a viable process. It's going to be a novel baby. But there are a lot of ethical issues associated with something like this. It just questions the logic that should we be doing something like this. We'll talk about the ethical issues also around this later on. Okay? Let's just go through the subjects. That's going to be the last part of our class. Whatever are the issues with all of these. The next thing is your gene editing. Again, it's a similar term. The concept you have somehow followed under all these things. Again, same चीजें जो हमने पढ़ी हैं, you can write down in one line. It is the type of genetic engineering in which DNA is inserted, deleted, or replaced in an organism. ठीक है. Again, जो अभी हमने concept पढ़ा वही है कि there is an organism, we have identified there is one gene. Suppose there is a gene which is causing a particular disease in an organism, and what we decide, we pick out those cells, we cut that gene from that particular site, we insert a normal gene in that, and then that organism starts functioning normally. So when we are using this technology for medical application, we also use the word gene therapy. But this gene therapy, for specifically when it is being used to create certain desired traits in an organism, right? Like concept of creating maybe a baby with a higher IQ, right? That is where this has come into question, or this has come into news in a lot of places because of its controversial. Abhi China mein somebody has tried to produce a baby like this with using this tool and with using CRISPR. You can write down about CRISPR also here. Clustered regularly interspaced palindromic repeats. So this is a set of molecules. It has two components. एक तो इसमें क्या है? इसमें RNA है. ठीक है. Single RNA is always single stranded. Okay. This is there along with a nuclease, an enzyme which is going to cut the things. Cas9. You remember what was a nuclease? Nuclease क्या था? Endonuclease. अभी थोड़ी देर पहले हमने बात की थी. What is a nuclease going to do? Cut. ठीक है. This is the molecular scissor which is going to cut, and this CRISPR is actually known to have both the elements together. It is going to cut at specific sites and it is going to heal and seal the DNA also. ठीक है. You just need to know the name of this, and because it has been there. So इसमें repair mechanism भी है साथ में. ठीक है. Cutting का भी है, joining का भी है, repair का भी. Everything is together in this. So what this is doing is the presence of, इसको single guided RNA भी बोलते हैं इस केस में. Sorry. So we need something to identify the DNA places also. So this single guided RNA, it goes over the DNA molecule and it identifies the specific site. Once that specific site is identified, this nuclease is going to cut it. So this one component CRISPR is doing two jobs together. That is why it is very easy to perform gene editing by this tool. ठीक है? Is this okay? CRISPR, it has two components. It has an RNA component and it has a nuclease. RNA will help in identifying specific sites. Okay. And this Cas9 is the nuclease which is going to cut the DNA at specific sites. ठीक है, सो जीन हमने आइडेंटिफाई कर लिया, काट लिया, उठाया, कहीं और ऐड कर दिया. Gene editing in this process. Now, one more conceptual idea you have to see. ठीक है, somatic cells and your sex cells. If we are editing a gene in your somatic cell, ठीक है, suppose you have a deficiency of pancreas, your body does not have pancreas genes, I insert that gene for production of insulin in you. It stays with you and the process ends here. But if I make a change in the genes of your ovum or your sperm or any of the sex cells, that means it is going to be now inherited. ठीक है ना? Because now it goes to the next generation, it does not stay here. Gene editing, whichever cells it is done in, that is what determines what its end is going to be. If it is done in a somatic cell, that means it ends there in that generation. The expression is going to be there only in that individual. But if it is there in a sex cell or in a sex gamete, that means it will be transferred to the next generation also. Okay, you are following this concept? ठीक है ये? ओके, ये सब आप उसमें भी क्या उसे बोलते हैं, करंट अफेयर्स में भी ऐसा कुछ कुछ यू विल कीप रीडिंग फ्रॉम टाइम टू टाइम. Okay, let's talk about one more concept before we can talk about the overall usage and application of this. This last concept for you here, stem cells. Okay, so we understood the concept of first formation of a zygote. What is your zygote? How is it formed? ठीक है, fusion of your sperm and ovum, it leads to formation of zygote. Now this zygote will have the capacity of forming the whole individual. That means this cell can divide, and not just divide, it can take the form of all the cells. ठीक है, from this only there's going to be a muscle cell, from this only there's going to be a blood cell, there's going to be a nerve cell. It will be able to, this one cell changing into another cell, we call it differentiation. ठीक है, the process is differentiation. So this one zygote cell, this is a single cell. This starts as a single cell, right? This single cell can divide and differentiate into all the cells of the body. This tendency this tendency of a cell to change into all the forms or all the cells is called totipotency. So totipotency will be one cell changing into all the different forms of the cell. Not just embryonic. अब ये zygote जो है, it will not just form the embryo, it will form all the cells and tissues surrounding it also. Like there is an embryonic fluid, there is placenta, right? When the baby is born. All those cells, this totipotent zygote will be able to form. One more concept is there, which we say pluripotent. A pluripotent cell will be able to differentiate into all types of cells other than your extra embryonic cells. Okay. So that means this is also dividing into everything, but जो extra embryonic structures हैं, like the embryonic fluids and placenta, that it will not be able to create. मतलब body में if there is a cell which has the capacity to divide and differentiate, it is going to be pluripotent. This only in the embryo. ठीक है. Stem cell इसी तरह से क्या है? Stem cells are those cells which are unspecialized, undifferentiated and have the capacity to divide and change into different forms of cells. You can just write down. They are unspecialized cells which can divide and grow into different types of cells. Now, types. Embryonic stem cells in the embryo, starting with the zygote. Here we are going to have both pluripotent and totipotent cell, right? Then the second one we have your adult stem cells. That means there are certain cells in the body which are still not specialized and which can be divided into other cells. Adult stem cells are found in bone marrow, skin, blood, parts of brain and spinal cord. Bone marrow, skin, blood, parts of brain and spinal cord. Brain and spinal cord. Yes. You don't have to know the parts. Then the third one can be induced pluripotent cells. They are your reprogrammed cell. Where are blood cells formed in the body? सही बोला है, कॉन्फिडेंस रखो थोड़ा सा. Huh? From stem cells, yes, obviously. But which part? From bone marrow? Remember we studied something about at least one cell, so particularly we studied lymphocytes, B lymphocytes. Where are they formed? Okay. अब देखो, embryonic will have the capacity to change into any type of cell. ठीक है? Because from this the individual is growing. Like now there is a concept of saving these embryonic fluid or stem cells. If you have heard कि from that placenta, from that umbilical cord, they are storing whatever amniotic fluid they are getting and storing the cells. ऐसा कुछ ad वैड देखा किसी ने? Stem cells save करें. एक ऐश्वर्या राय का भी ad आता था. हाँ, उसके लिए पे करना पड़ता है. So what happens is that at this stage, when a baby is born, whatever stem cells are there in that umbilical cord, they conserve it, they save it under cold temperatures if there is a need for that baby or his family to use the cell at a later stage. Advantage क्या है? These cells can be used to form into any type of cell. ठीक है? Again, that common disease which we've been talking about, let's consider that only. If there is some problem with the pancreas of this baby, जिसका हमने embryonic cell, embryonic stem cell we have preserved. What we can do is, we can take those cells, we can grow them to become pancreatic cells, we can introduce them in that baby, and that patient suffering from diabetes will no longer have diabetes because we have induced those cells. Similarly, it can happen for any other part of the organ. Suppose there is a deficiency of growth hormone. इनसे individual भी full बन सकता है. Yes. Again, through the process of cloning, but that is not how we are going to be using it. We're just going to take them, try and convert them into any specific type of cell. Suppose there is yes, heart disease also, any form of disease. Suppose there is a blood disorder, there is deficiency of certain types of cells which are not being produced or which are being produced in excess, any of that problem, we can alter that from this stem cells which we already have, we can induce them to become any type of cells. Organ develop कर सकते हो आप पूरा organ भी develop कर सकते हो, part of that organ भी आप develop कर सकते हो, क्योंकि ये जो cell हैं, ये इनके पास ये potential है कि आप इसको किसी भी form में change करो. The other advantage is it's not just for that particular patient, even for that family, right? Like you take an organ from a different individual, ठीक है? From a donor, ठीक है? I need a kidney, I get a donor which matches somehow, blood group matching किया. उससे better है if I have stem cells of maybe my child, okay? There are those stem cells, so that has a greater possibility of matching with me, right? Because we share DNA, right? So even if I need a replacement of a kidney or some part of my kidney if it is not functioning, I can use those stem cells, we can change it into kidney cells, and that can be a form of regenerative medical therapy for me also, right? You're able to understand? इसमें ये है कि advantages, they are non-specific, so all types of cells they will form, right? The adult stem cells जो हैं, they are specific. So if I am taking it from the bone marrow, so I will be able to produce only those things which the bone marrow can produce. Okay. So if there is a deficiency, like a lot of times, in a lot of shows also they show कि the sibling bone marrow will be needed. कि इस बच्चे को कैंसर है, अब इसका भाई चाहिए एक जिसका बोन मैरो निकाल के इसमें डालेंगे, ठीक है? So the point is whatever deficiency is there, whatever lag is there, that particular, जैसे हमें blood cell बनाना है, तो हम bone marrow से वो cell लेंगे जो actually who are determined to make blood cell, we'll form those cells and then induce it in that person or in another सिबलिंग और ऑर्गेनिज्म विच हैज लाइक कॉमन डीएनए और शेयरिंग, तो उसमें हम ये यूज कर सकते हैं टेक्नोलॉजी. Instead of giving a medical treatment, instead of giving a chemical treatment, we can actually correct the basis of the defect here. Induced pluripotent cells में क्या है? Any normal cell which is not a stem cell, like your skin cell. अब skin cell has reached its destiny. ठीक है? It has become skin cell. It has no potential to divide or change into any cell. There is a possibility that we induce it back, we reprogram it to become pluripotent again. This is also the latest trend which is now possible. कि जो स्टेम सेल्स हमारे पास हैं बॉडी में वो तो ठीक है, अगर एम्ब्रियोनिक हमने सेव किए हैं वो भी ठीक है, बट हम नॉर्मल सेल को भी कन्वर्ट कर सकते हैं टू एनी टाइप ऑफ सेल्स वी वांट. Right? Do you understand these three types? So according to this, they can be utilized basically in your clinical treatment also and for the research perspective also, right? Like I want to develop a medicine for maybe some disease of the brain. So what will I do? I'll take a stem cell, I'll change it into a brain cell and I'll see कि मेरा जो medicine है वो कैसे act कर रहा है. What are the changes? I want to do that change. I want the research to be realistic on real cells. I can do that. I can study the impact like that. ठीक है? इस तरह की जो टेक्नोलॉजी है for studying, for treatment of disease, we can use them. इस पे questions UPSC has asked in prelims also, in mains also. So this is an important concept. The basic thing you want to know or you should be knowing is that this is a cell, any type of cell, which we can change into any other type. Divide होगा, differentiate होगा, जैसे हम चाहेंगे वैसे हम इसको कर सकते हैं. Now in general about this whole thing, let's talk about what are the challenges we have and what are the uses we have of biotechnology as a whole and any other issue. अगर हम पूरा सीक्वेंस करें, have you heard the word genome? What is a genome? If I say human genome, what do you think you will understand by that? Okay. If I have to make an account of all the genes of an organism, that becomes that organism's genome. In one organism, all the genes, that means the sequence of its whole DNA, a complete set of DNA of an organism with all its genes is the genome. Okay. So when we say like there is this human genome project, what do you think will be the role of this project? क्या करेगा ये? सारे genes को identify करेगा that in human beings what all genes are present. A project to understand that is going to be your human genome project. अभी government ने एक नया project start किया है इंडिया में, that is your microbiome, human microbiome project. What do you think that will be? थोड़ा सा दिमाग लगाओ. ये तो हो गया ह्यूमन जीनोम प्रोजेक्ट, आइडेंटिफाइंग द जीन्स ऑफ ह्यूमन बींग्स, ऑल ऑफ देम, कि विच जीन्स आर प्रेजेंट इन ह्यूमन बींग्स. व्हाट कैन बी दिस? What do you understand by microbiome? थोड़ा सा वर्ड को ब्रेक करके दिमाग लगाओ. of little bit connection of the biome. Okay, try and associate this with microorganisms. Of all the microorganisms connected with human beings. No? This is going to be a pool of all those microorganisms which are affecting the human beings. ठीक है? Pathogens, remember? ऐसा कुछ पढ़ा था हमने? ठीक है? So all those microorganisms which are impacting human beings, if we try and identify the sequence, their sequence, it becomes the human microbiome project. We are doing it. This is a new concept now. What do you think? How will this help? क्या फायदा, क्यों कर रहे हैं इतनी मेहनत? Yes. So basically, we are basically just trying to understand कि what is causing diseases. ठीक है? Even in this aspect, we are trying to identify कि what are the relationships between different human beings, different races, different segments. How are they related? And when we are trying to identify the microorganisms, we are just trying to study the diseases better. ठीक है? कि क्या diseases हैं, क्यों हो रही हैं, which are the genes which are causing, and once we know the cause of them, we will be able to find the solutions also to them. This is a different concept. This is a different curative, preventive. दोनों तरह से use कर सकते हो. Cure कुछ नहीं है. This is just a study. This is just a study of data. On this, based on this, if there is going to be an analysis. Okay. So think about biotechnology in general. Okay. Let's just try and identify कि क्या इसका use हो सकता है in different sectors. Okay. What do you think can be the use of biotechnology, or how you can find out anything from all aspects, all types of biotechnologies overall? Okay. Agriculture में कैसे help कर सकते हैं हम? एक करके. What will be the advantage of a hybrid plant? क्या क्या application? Okay. तो इसमें you can say increased productivity of crops. Okay. The next thing we can maybe try and achieve is pest resistance. Okay. We can have addition of nutrients. Okay. What else? Food security, yes. Through increased productivity, we can ensure that there is food security. Gestation period. Okay. So you can maybe. Okay. So we can have better returns through shorter growing periods. We can have crops which have shorter growing seasons. What else can you think of? Okay, so we can also relate this concept with our environment. We can say we can have crops which are adapting to your climate changes. ठीक है? Better adaptation of climate change and things like desertification, changing water levels, water tables. Okay. वो इसमें भी आ जाएगा. Then you can have you can relate it to pest resistance also. You can relate it to this water also, you can have a lower cost of inputs. Like you can have crops which need less fertilizers, less pests, so that actually reduces the overall cost of your crops. ठीक है, पानी भी हम ऐसा क्रॉप बना सकते हैं which don't need a lot of water, so that means irrigation cost is reduced. So input cost overall can be reduced. Yes, you can say that also, but on the whole, that's not a very sustainable practice. You know, growing crops from other regions, it's not very healthy as such for the ecosystem on as a whole. So, I guess this is it. Yeah. So UPSC has asked this type of a question also this time. They've asked, how does biotechnology change the or help in the improving the living standards of farmers? Right? So you can dwell on points like these with specific example and just try and write this answer. Okay, you just see this paper 3 में ऐसा कुछ question है. Now come to other fields other than agriculture. हाँ, मेडिकल में तो अभी बहुत सारी चीजें हमने पढ़ ही ली. क्या क्या कर सकते हैं? जीन थेरेपी है ना, जीन थेरेपी हो गया. देन वी कैन हैव रीजेनरेटिव मेडिसिन. व्हाट एल्स? स्टेम सेल्स इसमें ही आ जाएगा सबके अंडर. चलो, स्टेम सेल्स, स्टेम सेल थेरेपी. Something like a research perspective can also be helpful in medical biotechnology, that through research we have a better understanding of all our diseases now because of this. ठीक है? And our mechanism to tackle diseases in that sense is also improving. इसमें एग्रीकल्चर में आप ये भी वो डाल सकते हो, एक चीज और, लाइफ स्टॉक. जो एनिमल्स हैं, दे आर ऑल्सो अ पार्ट ऑफ योर एग्रीकल्चर. सो इम्प्रूवमेंट ऑफ लाइफ स्टॉक इज ऑल्सो वन एस्पेक्ट. लाइक वी कैन हैव milching animals which are giving us more milk, or maybe more nutritious component of milk we can produce by some basic genetic modification. Yes, you can say that, but again the point is कि इसमें zero budget में genetically modified organisms we don't include because वो हम क्रॉप में ही बना देते हैं. एक मिनट, तो उसमें बिकॉज देर इज एन इनिशियल कॉस्ट एसोसिएटेड विद क्रिएशन ऑफ द क्रॉप, तो यूजुअली वी डू नॉट ब्रिंग दैट अंडर दिस दैट टर्म. Yes, livestock improvement में भी again it is because it is genetically modified, so we don't exactly club it with that. Cattle improvement is there, but see उसमें improvement के there are two forms. If we are doing it by hybridization without changing their genetic makeup, ठीक है, then that can be a part, you know, that you are just taking those specific varieties and breeding them together and then going it. But अगर हम gene modification कर देते हैं उसमें नहीं include करते हैं. So what were you asking? हाँ, इसमें what do we do? We insert a pest resistance gene in this organism itself, so we don't have to give out pesticides from outside. ठीक है. अच्छा, इसकी एक ये भी बात कर लेते हैं जेनेटिकल मॉडिफाइड क्रॉप्स की ही. So what we are doing, like I'll take the example of BT cotton. ठीक है, BT cotton में what are we doing? We are inserting a gene which is resistant for pests. So we don't have to give pesticides from outside to this crop. Advantage क्या है? our cost will be reduced. This will give you greater yield because less of it is being spoiled and attacked by that particular pest. But what is the problem? अभी तक हम ये food crop में incorporate नहीं कर पाए. So we are not going to eat cotton. ठीक है? What are the challenges in doing that or in the use or promotion of these genetically modified crops? So this should also have been something which you have read about. What do you think can be the challenges of having food crops which are genetically modified? Okay, so what happens is I have a BT cotton, ठीक है, crop. I take a small field, ठीक है, जिसमें I am growing this. There is a field adjacent to it which has other wild varieties or normal varieties of cotton. But pollination is taking place. Okay, from this plant, your pollens will go into their plants. From there, it will come to these plants. So when this pollination is taking place, so those plants also, the wild varieties, they also start getting this gene or this component in the next generation. ठीक है, जब वो reproduce करेंगे साथ में तो that possibility will be there. So subsequently, that natural diversity, that natural biodiversity of that region, that will be disturbed. वो जो original composition है उन plants की, that will also be disturbed. So that loss of biodiversity is one factor. Then the next thing is that there can be some possible health concerns. ठीक है, loss of biodiversity, biodiversity is always natural only. पहले हमने तो एक बनाया पर हम ये नहीं चाहते कि दुनिया से सारा ही खत्म हो जाए ना, राइट? तो वो बाकियों को भी इन्फ्लुएंस करेगा, विच वी एक्चुअली डिड नॉट इंटेंड टू ऑल्टर. वी ऑल्टर्ड अ वेरी स्मॉल सेक्शन फॉर प्रोडक्शन, बट नाउ इट इज इम्पैक्टिंग आवर वाइल्ड वेराइटीज ऑल्सो. ठीक है, if we are consuming these, there can be some, because these genes are going to express in a lot of different ways, so we don't know what health concerns are there. ठीक है, it is still in the form of research, a very exact data. Like in BT brinjal, there was a problem कि it was having some gastric challenges, कि it is not being normally digested by people. So इस तरह के कुछ challenges भी इसके साथ हो सकते हैं. Then there is this concept of terminator seeds. What is this? So normally कोई भी plant होता है, next generation क्या है, उसी plant में से you get seeds, you sow those seeds and the next generation starts. In these genetically modified crops, what is the problem? How they have been created is that they have terminator seeds. These seeds which are produced from these crops, they are not viable for production again. This is a termination stage. So the next time you have to go and buy those seeds again. ठीक है? So the companies who are making them, like इस case में जो सबसे famous company है, that is Monsanto. ये सब आप वैसे current affairs में पढ़ोगे. इसने seed ऐसे बनाए हैं कि वो terminator technology use कर रहे हैं, so they will not be able to grow. So even if productivity increases in one crop, the farmer has to again go and buy it again. ठीक है, जबकि अगर वो वाइल्ड वेराइटी यूज कर रहा था तो वो वही सीड एक बार खरीद कर दोबारा दोबारा उसको ग्रो कर सकता है. हाँ, तो ये जो है, दे आर ट्राइंग टू क्रिएट मोनोपोलीज इन दिस सेंस कि आपको हर बार हमसे सीड खरीदना ही पड़ेगा. देयर इज नो अदर चॉइस यू हैव. ओके, एंड अगेन if in this process, if the microorganism gains resistance, resistance भी तो हो सकता है ना उसके बाद. ठीक है, so अगर resistance हो गया तो then this crop is again ineffective, and you with that belief कि भाई pesticides are not needed, you are growing this crop, but the crop is attacked with maybe a different microorganism. ये तो आपने जो gene use किया, that will probably be applicable to one particular type of pest, but what if a different pest attacks, then it becomes unviable. So these will be the issues, and you can also put them as issues अगेंस्ट और चैलेंजेस ऑफ बायोटेक्नोलॉजी एट लार्ज. ठीक है, 2 पॉइंट्स यहाँ पे जो होंगे, एक तो ये होंगे कि loss of biodiversity, this will be applicable for all forms of biotechnology. ठीक है, the second one is creating of monopolies, and the third point which is also relevant everywhere is that the economic divide which this can create. How this can create an economic divide? Countries, nations or segments of the society which can afford these genetically modified products, they will have a different progress record, and the others which cannot, they will have a different. So it just widens that economic disparity. Okay. So, or other aspects again in utilization, you can say for environment, you can say bioenergy production. ठीक है, we can have certain microorganisms like algae can be used for production of biodiesel. ये सब आप पढ़ोगे, environment पढ़ा आपने अभी? So इसमें ही पढ़ा होगा ना, biodiesel, bioethanol, bioremediation. Have you read about these concepts? याद नहीं है, पढ़ा है? ओके चलो. So इस तरह की applications भी आप add कर सकते हो इसमें to give a comprehensive idea of this whole thing. Okay, so if I ask you what are the challenges of maybe executing or promoting biotechnology, what do you think will be the challenges if you have to answer that? Challenges in the application of biotechnology. Okay, if I ask you challenges in the application of biotechnology in India particularly. Lack of? Okay, सबसे पहले पैसा नहीं हमारे पास. हाँ, what else? Ethical issues पे आएंगे एंड में. ओके, चलो रिसर्च भी उस लेवल पे वी हैव नॉट बीन एबल. वैसे बायोटेक्नोलॉजी में वी आर एक्चुअली रैंकिंग वेरी वेल एज कम्पेयर टू अ लॉट ऑफ कंट्रीज, स्पेशली जो फार्मास्यूटिकल सेगमेंट है हमारा. ठीक है, जिसमें हम जैसे इंसुलिन क्रिएट कर रहे हैं या ग्रोथ हार्मोन बना रहे हैं या वैक्सीन्स, अ लॉट ऑफ वैक्सीन्स आर बीइंग क्रिएटेड बाय बायोटेक्नोलॉजी. So in all those segments, we are actually way ahead than a lot of other countries. But still, the ground level, we can maybe have more research, more trained manpower, right? कोई भी जो भी टेक्नोलॉजी होती है उसमें एक चीज जरूर फीचर हमें ऐड कर सकते हैं इजीली. कहीं पे भी जिस मर्जी टेक्नोलॉजी के बारे में आपसे पूछ लिया जाए, one point you can always write is that there is no integration between research and industry. जो रिसर्च हो रहा है that is limited to your university, it is not going to the manufacturer who actually has to use this. This is a common point. आपसे robotics के बारे में पूछ ले, आपसे नैनो टेक्नोलॉजी के बारे में पूछ ले, आपसे जनरल इंजीनियरिंग बेस्ड रिसर्च, कुछ भी, ये पॉइंट आप जरूर लिख सकते हो कि रिसर्च अगर हो, पहले तो हो ही नहीं रही, अगर हो रही है तो दैट इज नॉट रीचिंग योर इंडस्ट्री. ठीक है, देन अगेन awareness level about these things is not that easy or not that prominent. Another facet you can add here is your intellectual property right. So there is a person in this class who has a problem with this not being taught, right? अच्छा आप हो? Okay. हाँ? क्वेश्चन आया था, कोई बात नहीं, आएगा, इसको पढ़ो. यहाँ पे भी इसको यूज कर सकते हो कि इंटेलेक्चुअल प्रॉपर्टी राइट्स कैन ऑल्सो बी ऑफ कॉन्फ्लिक्ट विद एनीबडी हु इज डेवलपिंग दिस काइंड ऑफ अ टेक्नोलॉजी. एंड इफ यू आर डूइंग एवरीथिंग ऑल्सो, देन अगेन मार्केटिंग एंड सप्लाई चेन मैनेजमेंट again is a challenge of replicating this on a larger scale. Then again last में एक चीज और, अब इतना नुक्स, हाँ, yes, this is the last aspect which you can add कि always अब सारी गलती कि कितनी अब इंडस्ट्री यूनिवर्सिटी की निकालें, थोड़ी सी सरकार की भी निकालते हैं, that regulatory channels, laws are not well defined and regulatory authority is needed for this. Again, towards the end of it, or any of these stem cell, cloning, gene editing, babies, one very dominant aspect is your ethical issues. What do you think is the ethical challenge? I want to have a baby with blue eyes, so I want कि इसका अब gene editing करके मैं ढूंढ के निकालूंगी, कैसे करना है? What is the problem with that? Huh? Domination of? Yes, this is actually a very abstract, but it is a very important point. You will find it to be written in a lot of places. They say कि whomever you are editing, that person has no say in that decision. Okay, so I am creating a baby with blue eyes. The baby might like green eyes. He'll grow up and tell me कि how dare you give me blue eyes? ऐसे कैसे? What were you thinking? So, yes, the consent, informed consent, usually whenever there is a treatment or a therapy which we are giving, so we use the concept of informed consent. So what is informed consent? That I am going to perform a surgery on you. I am going to ask you कि भाई ये surgery है, this is the procedure, how it is going to take place. I give you all the details, all the modalities. You decide if you want it. In this kind of a situation, informed consent is not possible, right? Because that life form is still not being created. So informed consent in any form will not be applicable here. Then again, the concept of maybe haves and have-nots. That the rich people or the affluent sections of the society, they will be able to have access to such thing. So imagine the gene editing has been developed to that level that none of the rich people will fall sick ever. They have all the best genes, they have increased immunity, everything. So their gap with those poor people, initially it's like at least disease is an equalizing factor, right? It does not discriminate if you are rich or if you are poor. So with this, that gap is going to widen. If we have that kind of a modulation, maybe for IQ if people start doing. So all intelligent people will be rich, they'll continue to become more rich, and the poor, without the access of such things, that disparity will grow. So this is another ethical conflict. Yes, this can also be one point. What is natural? Yes, तो natural को ही तो हम change कर रहे हैं इसमें, और कर क्या रहे हैं? No, we can change. Genes, if we are able to alter genes, right? See, again, feasibility of all these things, we are discussing this at a conceptual level, assuming कि yes, this will happen and this can happen and the possibility of developing this is there, right? अब feasibility तो ऐसे है कि see, like IVF, you understand what is IVF? In vitro fertilization. I take an egg, I take a sperm, I give it the best nutrients, I give it the most ideal conditions it needs to grow, then also it does not have more than 10% chances of growing. Okay, right? So these natural things will have their own mechanism. We cannot actually ensure everything in the lab. This is just a conceptual argument we are making कि अगर ऐसा हुआ, ठीक है, तो how it is ethically not correct. You can have something like this or to write about a concept like this in your ethics also, ठीक है? Ethics in medical science or ethics in this kind of genetic modifications, right? So there you should have these ideas to write this, okay? Yes, again natural flow जो है society का, this is a religious argument also. So a lot of religions, like specifically the Catholic Church is very prominent against all these alterations being done. They say that you cannot alter with what naturally God is giving. It is unfair. If there is a baby which is destined to be born with a disease, then that is how the baby should be born. You cannot alter the course of God. They are so very rigid against a lot of concepts. Even concepts like abortion is something which they do not appreciate, because they say that you are altering with the natural ways of God. So this is a religious argument also, right? And again, like one more point we had is that we are actually, with the loss of biodiversity, we are creating homogeneity in this society. We are only trying to pick out the good traits and replicating them, जिसको हम बोलते हैं designer babies. This formation of designer babies is actually a contrast to the fundamental natural lineage or inheritance concept. Okay. Anything else you can think of? Yeah. He's treated as a? He's not treated as a soul. Okay. What is he treated like? Yeah. खुद से जब वो उसी के अंदर बॉर्न होगा, हाँ फिर okay, this is so you're trying to say that if it is a surrogate baby being born, so that baby does not have a soul, not treated like a soul, ऐसा कुछ? I'm following it right? ऐसा ही कुछ बोला ना आपने? Okay, so I don't understand how. I mean, this is like too metaphysical for me to discuss or even to understand. Okay, we are not going into this, that when the soul is coming, it's going to become too spooky for me also. कि कितने हफ्ते में कौन सा soul आ रहा है, कौन सा नहीं आ रहा. I mean, imagine there's a baby and you're telling me there is no soul. Okay, this is an entirely different zone. Okay. Please don't write such things in a UPSC answer. The concept, unless and until you have a quoting from a very legitimate source, ठीक है? तो अगर पोप ने ऐसा कुछ बोला तो लिखना, otherwise please don't write such metaphysical things कि this is against the concept of So how we are treating a person? How are we treating a person? We are not treating a person as a person, we are treating it as a commodity. ऐसा कुछ? Yeah. Maybe you can write something like this. Because we are trying to do a lot of this, you have to still present it a little more objectively. I get that. You can use this, that when we are having a concept of something like a designer baby, we are treating a living entity as a product, as a commodity, and trying to do customization in that, right? ये लिख सकते हैं, सोल मत लिखना प्लीज कहीं पे भी, एटलीस्ट साइंस के पेपर में तो मत लिखना, वो टीचर शॉक में चला जाएगा कि भाई क्या कर रहा हूं मैं ये. Okay, so I guess this is it. Try and build on all these issues. ठीक है, जो भी आते रहेंगे इसमें, जो ये वाला एस्पेक्ट्स है सब चीजों का, एडवांटेजेस, डिसएडवांटेजेस, दैट यू हैव टू नो स्पेसिफिकली. सुन लो यार 2 मिनट और, आखिरी क्लास है, कोई बात नहीं. ओहो 5 मिनट ज्यादा ले लिए मैंने आज, सॉरी. One more question I was asked in an earlier class. बिहार and लीचीज, पता नहीं किसने पूछा था, but yeah, मुजफ्फरपुर. Let's talk about that also. चलो बाद में, बाकी बाद में खेलना. Okay. Lychee. So you know about this case in Muzaffarpur which happened, 150 children died. What was the problem here was कि लीची में एक एजेंट होता है, ठीक है, स्पेशली इन दिस फॉर्म ऑफ लीची विच वॉज रॉ, जिसको हम हाइपोग्लाइसिन बोलते हैं, हाइपोग्लाइसिन ए. ओके, सो व्हाट हैपेंड हियर वाज दैट they were children. These children, what you have to know about them is that they were malnourished already. Malnourished means that they already had deficiency of food on a regular basis and nutrients. So you can associate this condition with a poverty-ridden household also. ठीक है? तो ये malnourished बच्चे थे. How is glucose stored in our body? Glycogen. ठीक है? तो glucose normally जो है, whenever it is in excess, it is stored in glycogen form, right? So this child, because it is already malnutrition, so glycogen reserves इसमें है नहीं. Okay. So this child does not have glycogen reserves. So how its body is functioning, whatever it is eating, it is being converted into glucose, it is utilizing it for energy. This is the normal course of metabolism of this child. Glycogen इसमें है नहीं. Now what has happened is that this child has eaten lychee and उसे लगा कि हाँ अब उसका पेट भर गया with this lychee, and this child goes off to sleep. Empty stomach because it has, he or she has eaten handful of these lychees. Now when the child is sleeping at night, अब उसका जो ये, इससे जो कुछ उसको nutrient मिला था, that is being utilized. The child is hungry. Now the body needs metabolism from another source. Glycogen आपके पास है नहीं. इसके बाद क्या चाहिए, क्या चीज होगी? What else can the body use once glycogen is not there? After glycogen, yes, fats. Now the body wants to metabolize fats or fatty acids. ठीक है? So normal process क्या है? This fatty acid we can eventually change into glucose and body can use it. Okay? ये जो agent है, this prevents this reaction. This hypoglycine A prevents fatty acids from being converted into glucose. ठीक है? ग्लाइकोजन आपके पास है नहीं, खाना आपने खाया नहीं, तो ग्लूकोज मिलेगा नहीं. फैटी एसिड से बना सकते थे, वो भी नहीं बना सकते. ठीक है? So there is a severe deficiency of any nutrient in this child, and because of that this child develops a disease of the brain. ब्रेन में इन्फ्लेमेशन हो रहा है जिसको हम कह रहे हैं एंसेफेलाइटिस, and eventually this child will die. Okay. What could have been done? This lychee intake should not have been empty stomach. After coming home, if this child who is already malnourished, if this child would have eaten something at home at night, then he would have had a normal supply of glucose throughout the night and would have survived. Or if this child was already not malnourished, ठीक है, अगर ये एक normal child होता आपके जैसा, ठीक है, जो मजेदार राजेंद्र नगर का बढ़िया वाला टिफिन खा रहा है, which has paneer every day actually, so then this will not be a problem because you have reserves of glycogen which will take care of you even if glucose is not coming in. ठीक है, फिर उसको फैटी एसिड तक जाने की जरूरत ही नहीं पड़ेगी. ठीक है, ऑल राइट. सो प्लीज कीप योर सेल्फ अपडेटेड ऑन करंट अफेयर्स. इन एनी केस इसके बाद भी कभी भी कोई डाउट आए, यू कैन ऑलवेज राइट इन योर टॉक टू द एक्सपर्ट कॉलम, राइट. करंट अफेयर्स इसका मोस्ट प्रोमिनेंट पार्ट है इन सब चीजों का. आई होप कंसेप्चुअली you are clear in all the ideas. Just build on current affairs from this area. Questions जरूर आएंगे, 2 segments पे ज्यादा focus करना है, health and diseases and biotechnology. The other things you just need for your concept. Science and tech का class हो गया आपका? Okay. So नीरज नचिकेता सर की जो book है उसमें basic science का भी segment है. So if any of you, if you have that book, you can go through that portion also from there if you intend for any specific parts. Okay, chalo, all the best.